webinar covers the topic of smooth muscle. During this lecture, we're going to predominantly be looking at the following things. We're going to be looking at where it's located, overall functions of smooth muscle. We'll be looking at different types. In one particular type, I will talk about a lot more this trimester and next trimester, and I'll make a point of telling you which one that we're going to be talking the most about. We will look at the anatomy of smooth muscle, and it's very important that as you're listening and taking notes, that you always try to see how it differs from skeletal muscle. So that's kind of a big thing to try to remember. We're also going to look at the various functional characteristics of smooth muscle, and again, try to remember how it differs from skeletal muscle. We're going to be looking at the excitation-contraction coupling, how it's similar, how it's slightly different than skeletal muscle. We'll be looking at the length tension characteristics in smooth muscle, energetics, in other words, energy involved in smooth muscle contraction, and also the control of smooth muscle contractions. First, let's look at locations. The smooth muscle is located predominantly or in most hollow organs, say like your stomach. It is along the GI tract, like the esophagus, the small intestine, the large intestine. It's in your urinary bladder, ureters. You'll see here in your bronchioles, blood vessels. It's also in the uterus. The myometrium is smooth muscle. So it is, think about kind of its function in, say, the stomach, the small intestine, the esophagus. It's involved in peristalsis, which is this rhythmical movement of, in this case, food along the GI tract. Say in the stomach, it's involved in mixing movements. It's also be involved in mixing movements along the GI tract too. It is going to be involved in moving blood through your cardiovascular system, allows you to maintain blood pressure, be involved in controlling airflow in the lungs, and think about what its role would be in the uterus, obviously in propulsion of a baby during parturition. So it serves a lot of different roles. You think mainly like skeletal muscle movement, but it also, I had mentioned before about mixing movements when we talk about digestion. We'll be discussing that next trimester in physiology too. It also, you'll see them in sphincters. Sphincters control movement of, or I should say entrance and exit of various things from the body. For example, you have a sphincter up here, sphincter here, which will control movement of fluid into the stomach and out of the stomach. You have an internal anal sphincter, which is made up of smooth muscle, which will be involved in defecation. But I want to kind of make a note right now that you have an external anal sphincter and an internal anal sphincter. The internal one is smooth muscle. The external one is made up of skeletal muscle. So a kind of good thing to kind of remember is the difference between skeletal and smooth muscle in regards to, is it a voluntary muscle or an involuntary muscle? Skeletal muscle is always under control from the somatic nervous system. It can be consciously controlled, which would be the external anal sphincter. The internal anal sphincter is under control of the autonomic nervous system, which would be involuntary. So when we talk about the defecation process next trimester, I'll emphasize again which sphincter is skeletal, which is smooth, and the significance of that. It's also, smooth muscle is along the fallopian tubes, which helps to kind of move the egg along, sperm up, so they kind of can meet and have fertilization take place, which does take place in the fallopian tube. Now, a couple things that we're going to spend a little bit more time that you see in your notes, so you're going to have to take some notes, is the types of smooth muscle. There are two types. There's a unitary, or they also call it visceral smooth muscle, and the other type is multi-unitary. They call it unitary is because this muscle acts as a unit together, as if it was just one muscle, versus multi-unitary, kind of separate type. The unitary smooth muscle is what we're going to be discussing predominantly in Physio 1 and Physio 2. This will be the type of smooth muscle that you're going to see in the bladder, in your arterioles when we talk about blood pressure control. You're going to see this when we talk about digestion in the GI tract. So this is the predominant type of smooth muscle we're going to talk about. The reason why it can act as a unit is it's going to be coupled. Each is going to be coupled together by gap junctions. And gap junctions allow the electrical impulse to be transferred from cell to cell very quickly, so the impulse travels throughout rapidly, and they will contract pretty much together and act as what we call a functional syncytium, as one. One thing to keep in mind is this type of smooth muscle does not exhibit recruitment. As we mentioned before about motor unit recruitment, this doesn't exist in the unitary smooth muscle. An interesting thing is that this type of smooth muscle exhibits spontaneous depolarizations. It has its control can be myogenic, which means it originates within the muscle itself. Skeletal muscle, remember, always needs, or is always told what to do. The somatic nervous system sends a wave of depolarization down and says, hey, you need to contract. Visceral smooth muscle doesn't need that. It can generate its own electrical impulse within the muscle itself. So this is going to be one way it can differ from skeletal muscle. Now this is unitary or visceral smooth muscle. The other type is multi-unitary. You're going to find this in the large airways in the lungs, the very large arteries, the eye muscles, the erector pili muscles, which are involved in like giving you the goosebumps when you're cold. These ones, they don't act as a unit, as one, because they don't have a lot of gap junctions. They don't usually have the spontaneous depolarization, even though they can, but they're not as frequent as you see in unitary smooth muscle. They do exhibit motor unit recruitment and will respond to nervous stimuli, and it can have the contractions graded as we talked about with skeletal muscle. You don't see that with the visceral or the unitary smooth muscle. This type of smooth muscle, though, we will not discuss very often. I mean, this is probably the only time I really discuss it. The unitary smooth muscle is what we'll discuss a lot more, and I will review it with you next trimester in Physiology 2. Now we have to discuss the anatomy of smooth muscle and how it differs so much from skeletal muscle. So here's some key things to remember. Why do they call it smooth muscle? The reason why they call it smooth muscle is it's not striated. The reason why it's not striated is it does not have a regular arrangement of actin and myosin. Actin and myosin are there. They do interdigitate, because you cannot, if they don't, you cannot form cross bridges and you cannot generate muscle tension, but it's not as a regular arrangement as you see in skeletal muscle. So we don't have what you call sarcomeres. They don't exist. So you're not going to see the striations as you see in skeletal or even cardiac muscle. There's a difference in the numbers of thick filaments. They have much fewer thick filaments than the thin, but what's interesting is they have a lot more myosin heads, and I don't know if you can kind of maybe tell here. It's a weird depiction of it, but the myosin heads along here go along the entire length of the thick filament. Remember, the thick filament is myosin, the thin filaments are the actin. The myosin heads run the entire length of the of myosin, and so you can have the smooth muscle, the contractions be just as powerful as skeletal muscle, because the tension comes from the myosin reacting with the actin filaments. So you can get very powerful contractions from smooth muscle. Now the filaments are arranged more diagonally, so you kind of see it here, they're running more diagonally. Because of this diagonal arrangement, that they kind of look, the contractile proteins crisscross, kind of like a spiral fashion along the long axis of the cell. And so when they contract, the muscle, I'm going to show you a picture, when they contract, it kind of makes a, it twists, and it looks kind of like a corkscrew because of the arrangement of the actin and myosin. But you also notice from this slide, you have these intermediate filaments in smooth muscle, kind of a lattice-like network of intermediate filaments that are contractile. And what they do, the function of them, is they help to resist tension, and they attach at regular intervals to these things called the dense bodies. So you can see the dense bodies here. They have in the blue. They attach to the dense bodies to kind of give a structural component to the smooth muscle. The dense bodies are also attached to the sarcolemma. Remember what the sarcolemma is. Sarcolemma is the fancy term for the smooth muscle, or in this case, sorry, muscle, the muscle cell membrane. So the dense bodies are attached to the sarcolemma. They act as anchors for the thin filaments. So if you kind of think back, try to like envision when we talked about skeletal muscle, the thin filaments were attached to something, but they were not attached to dense bodies. So what structure in skeletal muscle is somewhat analogous to the dense bodies that we see in smooth muscle? We'll give you a second to try to remember. It's got a letter. It's the Z discs. So in skeletal muscle, the thin filaments are attached to Z discs. In smooth muscle, the thin filaments are attached to these dense bodies. When we also look at the structure or the anatomy of skeletal and smooth muscle, I want to kind of compare these two. So it's always good to kind of remember, compare skeletal versus smooth. Skeletal muscle had T-tubules. What's the function of T-tubules? When the wave of depolarization came down from, remember, it originated at the neuromuscular junction, spread down through the T-tubules deep into the muscle. The T-tubules were invaginations of the sarcolemma, allowed us to transmit that depolarization wave deep into the skeletal muscle, where then it triggered the release of calcium, which was very important with muscle contraction. In skeletal muscle, the sarcoplasmic reticulum, which is a storehouse for calcium, are very well developed. Well, smooth muscle is different. Smooth muscle do not have T-tubules. Instead, they do have these kind of small invaginations of the sarcolemma that you see here. They're called, and this is spelled wrong over here. This is the proper spelling for calveoli. So that's a big no-no there. Calveoli. And what happens is that in response, for example, we're going to mention, like, if we do have innervation of the smooth muscle, that nervous input is going to release a neurotransmitter, which ultimately we're going to result in the calcium release from sarcoplasmic reticulum, but the sarcoplasmic reticulum in smooth muscle is very poorly developed. So you can get some calcium release by the sarcoplasmic reticulum, but Smooth muscle relies more heavily on calcium in the extracellular fluid, which, if you see here, this is part of the extracellular fluid. So what the alveoli do is they're going to sequester extracellular fluid that contains a very high concentration of calcium, and it keeps it very close to that membrane. So when calcium channels in the alveoli open, there's going to be a huge amount of calcium influx into the cell very quickly, which is going to allow smooth muscle to contract. Calcium is just as important in smooth muscle contraction as skeletal muscle. The only difference is, or not only, but a big difference between skeletal and smooth muscle is that smooth muscle relies more heavily on extracellular fluid levels of calcium than the sarcoplasmic reticulum, because it's not as developed in the smooth muscle. Skeletal muscle, its calcium comes completely from the sarcoplasmic reticulum. There's some functional characteristics between, or differences between skeletal and smooth muscle, and we'll discuss the slight differences as we go. We're going to be looking at the excitation-contraction coupling, and maybe in the process review a little bit of the skeletal muscle. We'll look at the length-tension relationships, energetics, and also the control of contractions, which is probably of the two of the if I'm going to say of these four topics, the two that are probably most important is the first one and the last one. So let's look at excitation-contraction coupling. Sliding filament theory. We had discussed that when we talked about skeletal muscle. Same applies with smooth muscle. You have the actin and myosin forming cross bridges. You have the pivoting of the myosin heads. That all still applies, but the difference is because of how it's regulated. In order to have skeletal muscle contraction, you had to have calcium bind to troponin C. When it bound to troponin C, you got a conformational change, moved tropomyosin out of the way. So then what happened? Think about it. More times you review this, more likely you're going to remember it. So let's think about it. Troponin C bound calcium. Conformational change moves tropomyosin out of the way. What's tropomyosin? Why do they call it tropomyosin? Remember, tropomyosin is part of the actin filament. Troponin and tropomyosin are part of the actin filament, but it binds to or inhibits the interaction of myosin with actin. But if tropomyosin's moved out of the way, there's now a site for myosin to interact with the actin, forming a cross bridge, and we can get contraction. Well, smooth muscle, troponin is not involved. Troponin complex is not involved in the regulation. Troponin will not be our calcium binding site. It's calmodulin. So when calcium is available, it will work via calmodulin to end up regulating the contraction. So it's going to be, let's go through this. This one's a little bit different. So what you see here is we're going to have calcium coming in from the extracellular fluid or with some within the sarcoplasmic reticulum. The stimulus for calcium release can be a number of different things, and we're going to discuss that later. So calcium influx, which is extremely important for any type of muscle contraction. What happens here is calcium will bind to, and this is how they abbreviate it, calmodulin. Calcium binds to calmodulin. There's 4 binding sites and calmodulin for calcium. When they interact, there will be a conformational change in calmodulin, which what it does is this complex, calcium calmodulin, becomes a regulatory protein for a number of different enzymes. And we're just going to be discussing one right now. there is an enzyme called, and this is how they abbreviate it, myosin light chain kinase. So we're going to do a little quick flashback back to biochemistry. In biochemistry 1, you should have learned a number of different ways how enzymes may be regulated. They may be regulated by reversible covalent modification, like does it have a phosphate attached to it or not? Is it regulated allosterically? If something binds to it, it changes the shape of that protein, changing the activity of it. So those are different ways that we can regulate enzymes. Another way is by what we call protein-protein interaction. So, calmogen is a protein. Well, calmogen is a protein that will bind to myosin light chain kinase only when the calcium is bound. So calcium is an allosteric regulator of calmodulin. This will bind to this enzyme, activating it. So we have activation of myosin light chain kinase by a protein-protein interaction. You should remember in biochemistry what kinases do. If someone says kinase to you, or if you see anything ACE, you're like, oh, it's an enzyme. Kinases' jobs are to phosphorylate things. In this case, this enzyme phosphorylates the myosin light chain, hence the name, myosin light chain kinase. So what happens is it will use the phosphate from an ATP to phosphorylate myosin. When it's phosphorylated, it is now active. This may now bind to actin, and you will subsequently get your cross-bridge cycling, and you'll get your muscle tension. So that after this point, it's going to be exactly what you saw in skeletal muscle. But the only way myosin can bind to actin is that the myosin light chain must be phosphorylated in order for that to happen. Otherwise, it won't happen. So, big difference here. Regulation of contraction of smooth muscle requires calcium calmodulin. Troponin is not involved. So, calcium calmodulin will activate the myosin light chain kinase. You did not really see that when we talked about skeletal muscle. That activates the myosin light chain kinase, which phosphorylates the myosin head. This will allow the myosin to bind to actin, and then everything else proceeds as you saw in skeletal muscle. If there are enzymes called, or an enzyme called myosin light chain phosphatase, phosphatases remove phosphates. So if the phosphatase is active, it will remove the phosphate, and then it won't be able to interact with the actin. Anytime, just like with any type of muscle contraction, if calcium levels are too low, we're going to start sequestering calcium in the sarcoplasmic reticulum using calcium ATPase or sodium calcium antiporter. We also can, the enzymes can be out here or on the cell membrane to get into the extracellular fluid. If the calcium levels within the cell drop, you will not have smooth muscle contraction. That same as what you see with skeletal muscle. Smooth muscle, like cardiac muscle and skeletal muscle, are innervated. It can respond to nervous system stimulation, but there'll be some differences. But before I go on it, I do want to emphasize that smooth muscle, unlike skeletal muscle, do not need the nervous system to have a contraction. It can be affected by a number of different things, but right now let's at least talk about the nervous system innervation. Skeletal muscle is innervated by the somatic nervous system. Smooth muscle is innervated by the autonomic nervous system. So there's a big difference there. In smooth muscle, you do not have true neuromuscular junctions, those very well-defined neuromuscular junctions that you saw in skeletal muscle. Instead, they have these things called varicosities. Varicosities are kind of these more or less kind of bulbous-like swellings that are filled with neurotransmitter. So these guys are filled with neurotransmitter. They release the neurotransmitter into synaptic clefts, but the synaptic clefts in smooth muscle are very wide. So they sometimes use the term a diffuse junction. So when the neurotransmitter is released, it's kind of more of a diffuse fashion. It's more a little bit more widespread and instead of being very specific. When the neurotransmitter is released, what is kind of a big difference between skeletal and smooth muscle, the effects can be excitatory or inhibitory depending on what the neurotransmitter is. And even when we talk about some other factors, some local factors or hormones, those things can be either excitatory or inhibitory on the smooth muscle. So it could either cause the smooth muscle to contract or relax. Skeletal muscle, it's only excitatory. You cannot have inhibition of skeletal muscle. So when the somatic nervous system does its thing, it's always excitatory. It is never inhibitory. In smooth muscle, it is either excitatory or inhibitory. And I'm going to give you some examples a little bit so you can kind of see how it's going to tie into what we're going to be doing later in the tri and next trimester. So let's talk about some hormones, or we can even talk about neurotransmitters. In smooth muscle, especially when we talk about the GI tract, they have some weird type neurotransmitters that, and I might as well mention now, in GI tract, they have an intrinsic nervous system. They respond not only to the autonomic nervous system, they have an intrinsic nervous system. And they have some slightly different neurotransmitters that you may not have realized. And we'll talk about them next trimester. But I want to mention some hormones. Like you said, well, norepinephrine's a neurotransmitter. Well, yes, it is. But it also is classified as a hormone because it can be released by the adrenal medulla, circulate in your blood, and have effects a large distance away. So the reason why I'm putting these all in here, I was like, oh, I might as well put these in because these are different factors that I'm going to talk about this trimester or next trimester, and I might as well have you kind of familiar with these right away. So you can have a hormone that can cause smooth muscle contraction, and that's somehow going to be something that influences, like you see over here, the release of calcium from the sarcoplasmic reticulum or through opening calcium channels in the sarcolemma, increasing the intracellular levels of calcium so you can get smooth muscle contraction. Or you can have a hormone that somehow will inhibit this calcium influx into the cell and would result in smooth muscle relaxation. I don't want you to start worrying about all these signal transduction pathways. You should have covered these in Biochemistry 1 when you should have talked about phospholipase C and cyclic AMP and cyclic GMP. I'm not going to emphasize that right now, maybe in class, but at this point in time, don't worry about it. I want to introduce you to various hormones that have specific effects on smooth muscle. So the first, I have to count myself, the first 5, these are examples of hormones that cause smooth muscle contraction. And I'm going to show you kind of where we're going to be discussing these. Norepinephrine, angiotensin II, endothelin, and vasopressin we'll be discussing when we talk about control of blood flow this trimester. So what these do is they cause vascular smooth muscle contraction. That's the smooth muscle along your blood vessels, which would decrease blood flow to a particular area in your body. There will be some of them not all of them, will be important factors or important regulators in maintaining blood pressure. But they're obviously involved in control of blood flow. So that's where we're going to be discussing these. You'll also be hearing these, norepinephrine, angiotensin II, when we talk about control of filtration in the kidneys by affecting the diameter of either the afferent or efferent arterioles in the kidneys. Oxytocin I just threw in because I'm like, well, think about this. Oxytocin is released, and it's released from the posterior pituitary. It's actually made in the hypothalamus, but stored and released from the posterior pituitary. This hormone is very important during childbirth. It stimulates very powerful muscle contractions from the uterus. The myometrium will contract very powerfully in order to get parturition or delivery of the child. It's also going to be involved in breastfeeding. It will cause contraction of myoepithelial cells around the mammary glands. It's going to be involved in milk letdown and milk ejection. So it causes contraction of smooth muscle. Nitric oxide and adenosine, we'll talk about this trimester and next trimester. Those will result in smooth muscle relaxation. Nitric oxide is produced by the endothelial cells, which line your blood vessels, and it's a very good vascular smooth muscle relaxer. So it would cause blood vessels to open up or dilate, which would increase blood flow. Adenosine is also a very good relaxant. This is a byproduct of the breakdown of ATP. If you take all the phosphates off of ATP, you're left with adenosine. We will be discussing a lot of these factors when we do our lecture on local control of blood flow. So you're going to see a lot of these again. Some other factors that are produced locally can control contractions. Some local ions, if you have changes in certain ion levels, that can affect whether or not the smooth muscle is contracted or relaxed, or even something like lactic acid. Lactic acid is a byproduct of anaerobic respiration or glycolysis, and lactic acid is a vascular, or it's a, I keep going to vascular smooth muscle because I'm so used to teaching about the cardiovascular system, is it causes relaxation of smooth muscle. Importance of that, say, when you're exercising. Producing lactic acid, you need to get rid of it, otherwise it's going to cause you to fatigue pretty quickly. You need to get rid of it. Well, that stimulates the muscle to relax, increase in blood flow to that tissue, which you want. You need more oxygen, you need to get rid of the waste products, you need to deliver substrates to use to make energy, and that's going to increase blood flow to that particular tissue. One thing that's important to realize too is the role of stretch with smooth muscle. And predominantly we're going to be talking about this very much next trimester when we talk about digestion and talk about, for example, movement of food through the GI tract. And stretch is a very potent stimulus for contraction of smooth muscle. So think about what would happen when you swallow. You chewed up your food, swallow it, hits the esophagus. What do you want? Do you want it sitting there? No, I want to move it down to the stomach. Well, that presence of that bolus of food is going to stretch the smooth muscle lining the esophagus, and that will trigger a peristaltic wave that will move the food down towards the stomach. Same will apply as help moving food down through the GI tract. Stretch is a good, it's a good thing that will control or stimulate smooth muscle contraction. You do not see that with skeletal muscle. Okay, so we talked about, like stretch reflexing things like that. It's like, well, if you stretch out a skeletal muscle, you get a reflex contraction of it. That's true, but only if you have innervation. If you denervate that muscle, that will not happen. I can denervate a smooth muscle and it will happen. It's intrinsic to the muscle. Same will apply with cardiac muscle. So when you talk about control of contractions, skeletal muscle, it's completely controlled by the somatic nervous system. Nervous control and its somatic nervous system. Smooth muscle, you've got an intrinsic system, you've got autonomic nervous system, you've got hormones, you have local factors, you've got stretch. So a lot of more different things can affect contractions of smooth muscle. Another difference is length-tension relationships. In skeletal muscle, there's a relationship between the resting length of the muscle and how much tension that can develop. Remember, if the sarcomeres are stretched too far, your zone of overlap will disappear or decrease. Your cross bridge formation will decrease, or it may not occur, so you're not going to get a lot of tension developed. If the muscle is too short, too compressed, you're going to get thin filaments extending over the center of the sarcomere, and it can interfere with normal orientation of the thick and thin filaments, which again would decrease tension. But in smooth muscle, it does not exist. You do not have that length-tension relationship as you do in skeletal muscle. I want you to think of the significance of this. Why is it so significant? Well, let's just discuss something first, and then maybe you're going to realize the significance or relevance of it. Smooth muscle, you stretch it, stretch it, stretch it. It will continue to have the same ability to contract if you ask it. And skeletal muscle will be like, your ability to generate tension is going to be diminished the more you stretch it. Smooth muscle, no. It exhibits what we call plasticity. I can't say it. Plasticity, which is an ability to function over a wide range in lengths. So you stretch it, stretch it, stretch it. It's still, if you ask it to, it's going to contract, and contract very forcefully. It will adapt to its new length. So when would this be significant? Think of your stomach when you're eating. You fill it up, fill it up, fill it up. It's not going to get that food out of the stomach right away because you need time to kind of start breaking it up and mixing the gastric juice in it. It's going to be sitting there, and if you eat a lot, your stomach's going to expand. You still want the stomach to churn that food and mix it up, so it needs to be able to do this. Even better example would be a pregnant female. Think about, as that baby's growing and growing and growing, it's stretching that myometrium out quite a bit. And when you need that baby out, you better be able to do very forceful contractions even though it's stretched so much. So that's why it's so significant. You don't have the same length-tension relationships. So in smooth muscle, you can still get very good tension development even with that muscle being really stretched out. You don't see that in skeletal muscle. Now the last topic that I'm going to discuss in regards to smooth muscle is having to do with energetics. I'm going to show you a little video. It's short. It's not really a video. It's a very short animation. Big thing it's kind of complicated, and they have theories about how this works. Big thing I want you to realize about smooth muscle is this first sentence here. Smooth muscle can maintain force for very long periods of time. It can maintain a contraction for a very long period of time and maintain tension with very little energy expenditure. So it doesn't use as much ATP as with skeletal muscle. Why is that so significant? Think about your blood vessels. Your blood vessels are in a chronic state of contraction. Your arterioles have a chronic state of contraction that allows you to maintain your blood pressure. You don't want to have to use a heck of a lot of energy to do that. And smooth muscle can do it. And think about when you're digesting your food. You need to slowly move it down. You need to do the mixing movements. And you don't want to have to expend a lot of energy to do it. So something about smooth muscle allows us to maintain a contraction with very little energy expenditure. And it's what they describe it as a latch state, or the latch state mechanism, whereby you can have the myosin and actin maintaining connections, being attached for long periods of time with less ATP generation or requirement. They say, I think they say, they estimate it's only like 1 ATP for every cycle, no matter how long the duration is going to last. The cross-bridge cycling is a lot lower, but it can still generate a good amount of tension despite the being able to be holding that cross bridge. It has something to do with the dephosphorylation of the myosin light chain by myosin phosphatase. But the exact mechanism is kind of uncertain. But so the biggest thing I want you to remember, this is what you don't have to tell me exactly the whole mechanism of how it works, but I want you to know the significance of this latch state, that you can maintain tension or force for long periods of time with little energy expenditure and smooth muscle. And it comes down that you have prolonged periods of attachment of it and without having to use a lot of energy. So let's see if I can get this thing to work.
This animation shows one of the ways that smooth muscle conserves energy, the latch mechanism. During each cross-bridge cycle, the inorganic phosphate can be removed at any point by a phosphatase, the gray box. Once the phosphate group has been removed from the light chain, the cycle will continue, but at a much slower rate of speed. In fact, it may stall at the point where the myosin head is bound to the actin active site and the ATP is bound to it but has not detached. As long as the active site and the cross-bridge are attached, tension is being developed. This is one way smooth muscle uses to maintain contraction for a long period of time without using much ATP. I should note for you that until another phosphate binds to the myosin light chain, a new cycle cannot begin again.
Let's watch it again.
This animation shows one of the ways that smooth muscle conserves energy, the latch mechanism. During each cross-bridge cycle, the inorganic phosphate can be removed at any point by a phosphatase, the gray box. Once the phosphate group has been removed from the light chain, the cycle will continue, but at a much slower rate of speed. In fact, it may stall at the point where the myosin head is bound to the actin active site and the ATP is bound to it but has not detached. As long as the active site and the cross-bridge are attached, tension is being developed. This is one way smooth muscle uses to maintain contraction for a long period of time without using much ATP. I should note for you that until another phosphate binds to the myosin light chain, a new cycle cannot begin again.
So you kind of saw that it was really slow. It stays attached, and it stays attached for a long period of time. It was really slow, but as long as they're connected, you can maintain tension. Once it's let go, though, like she said, it will not attach again until the myosin light chain is phosphorylated. Because remember, that's a big difference between skeletal and smooth muscle. The myosin light chain must be phosphorylated. So realize again, myosin phosphatase can remove that phosphate, but for some reason the actin and myosin stay attached, but the cycling is very slow. But as long as they stay attached, you can maintain tension. See if I can get this. Alright, so this is the end of the lecture on smooth muscle. You have another lecture on cardiac muscle that you must listen to also.